So somebody should listen to what I have to say, and they can call the other person back later, because what I have to say is super important, because I think I'm totally awesome. So in pediatric patients, about 60% enroll in trials. So why can't we get there for adults? Number one is nobody tells us about trials. When I was diagnosed metastatic, 5 years after my early stage diagnosis, when I had just written an article for my local newspaper on, yay, 5 years, I don't have to worry about this crap anymore. And then 2 weeks later, I was diagnosed metastatic on my birthday. Don't ever have scans on your birthday. After finally having symptoms and proceeding with scans that had metastases in literally every bone in my body, I asked the first oncologist that I saw, the one and only person who came to my small town 1 day a week and ironically sublet my office from me that 1 day a week. And I said, what about a clinical trial? You don't need a trial. You don't need a trial. Here's NCCN guidelines, which I don't think are right. Let's do this really crazy, aggressive combo IV chemo thing and keep you on it for, I don't know, like 14 months. I was actively discouraged from participating in a trial. Trials are often thought to be the last resort. You don't need a trial yet. We have all these wonderful therapies. Well, we wouldn't have all these wonderful therapies if amazing people did not participate in trials. And my opinion is every metastatic patient should be offered the option of a clinical trial upon diagnosis. Maybe not that first visit when the patient's freaking out, but maybe bring the patient back in. Maybe have someone talk about trials. Maybe some organizations could do a little video series that every patient could be educated about trials, because I know how busy you oncologists are. But let's leverage technology. Let's use things so that we can educate every single patient about the opportunity for a trial. Eligibility criteria are ridiculous. I call it, you have to be a cancer Olympian to participate in a trial, which means that the trialists want the healthiest of those of us that are dying with metastatic disease, because they want pristine results. I was just at the AAADV workshop where I was invited to be on a panel about decentralized clinical trials, and on a different session, an oncologist who now works for industry and is on one of the workshop panels about eligibility requirements, and specifically about, I think he was on the one about prior lines of therapy, made a comment that including patients who had had multiple prior lines of therapy was kind of like throwing dirt into a test tube. And all I can say is thank God Dr. Tatiana Prow of the FDA and Johns Hopkins got to the mic before I did, because she was much more polite in her response to that. When your drug is approved, believe me, there are going to be people on their 6th, 7th, 8th line of therapy using it. So we need to have eligibility criteria that reflect the population. There's also, from the patient standpoint, the fear that we may get a placebo in a trial. And here in the US, you don't get a placebo versus the study drug. You get standard of care. We're not saying here in the US at least, everyone gets at least standard of care. But why should I travel 4 hours to San Francisco to get something that I only have to drive an hour and a half to Truckee, California to get, because I don't even have an oncologist in my community? And so patients are reluctant to sign up for a trial for this very reason, because trials require travel for most of us. Not all of us live 5 minutes from a comprehensive cancer center. So there are travel costs, including hotels. If you're in a phase 1 study and they're doing pharmacokinetics on you and they're drawing blood every 3 hours, and you live 4 hours away and they want blood at 1, 3, 6, 9, 12, 24, and 36 hours, you have to stay there. And often those are costs that are not compensated, that are not covered by the study, and are certainly not covered by the insurance that we have. There's the time off work. You know, it's not like jury duty where you get paid at least, a couple bucks to participate in jury duty. No one's paying for that time you're losing from work. And if you're a younger patient and maybe you have childcare, or you're that sandwich generation and you're caring for elderly parents, then that is also a factor. None of those things are compensated, and those are all reasons why we have such abysmal rates of participation in clinical trials. So how can involving patients improve enrollment? Number one, your advocate needs to be disease and stage appropriate. As a metastatic breast cancer patient, don't put an early stage patient on a grant that is clearly designed for metastatic disease. Don't put me as a metastatic patient on an early stage grant, because I don't remember what it was like to be early stage, and the concerns of the metastatic community and the concerns of the early stage community are so different that I think it is incumbent upon researchers to seek out patients who are disease and stage appropriate. If you have an advocate involved, we're going to help you with the questions you're asking. I've reviewed some grants where, as someone who, like 100 years ago, was a biology major at Bryn Mawr, although I also majored in heavy drinking, so I'm not really sure I remember the biology part of it. But I understand the science a little bit, so I'll sometimes review a grant and go, oh my God, this is incredibly elegant science. But who gives a crap? Because this is not going to answer any question that is meaningful to a patient with this disease. But if you involve an advocate, then you are going to get better questions, meaningful questions. And if the endpoints and the questions to be answered in a study are meaningful to the patients, then maybe patients will be more likely to want to participate in your study. The design may be more palatable to patients. I reviewed a grant recently where I was asked to be the advocate, and I read the study design, and the study design was asking for tissue biopsies of metastatic sites at fairly frequent intervals. And I said, I called up the researcher, and I'm going, so why do you want to stick a big-ass needle in somebody's liver once a month? Well, we really want to start looking for these genomic changes, blah, 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 blah. And I'm going, have you ever heard of blood-based, NGS? Maybe do that. And are you going to change anything in your study design, in your protocol, based on these biopsy results? Well, no. Then don't do it. Sometimes put yourself in the shoes of the patient. If you wouldn't do it yourself, then don't ask the patient to do it. So they changed their design, and they asked for biopsy at diagnosis, at enrollment in the study, and at progression. So we took a study design that was going to require 6 biopsies down to 1, 2, possibly 3, just by asking a patient whether that would be acceptable to the patient. And then there's language. One of the things I kind of joke around about is that one of my greatest skills as a physician was translating medicalese into plain English. And part of that is I can't pronounce those big words like oophorectomy, much less spell them. So you translate it into terms the patient can use. I never said, I'm going to do a subtotal hysterectomy and a bilateral oophorectomy. It's I'm going to take out your uterus, your tubes, and ovaries. So we can do that in clinical trials as well. So to have the patient involved in reviewing your trial description to make sure it's understandable to the average person, and even things like the consent form. And yes, we have the lawyers, and we have to placate the lawyers, and they're going to want the 35-page consent form. Keep the 35-page consent form, but use your advocates to help you develop a 1-page, maybe front and back, plain language consent form that has the salient features, and you can put the little asterisk, please refer to our lawyer-approved consent form, so that you're fulfilling all those requirements without scaring a patient with a 35-page packet of information that they have to fill out. And also, if we're involved in your trial, in the design of your trial, we're invested in your trial, and we want your trial to succeed. And we can leverage not only our local networks of cancer patients in the community, but we can also utilize our very powerful social media networks to spread the word about the trial. And we can probably help you accrue patients faster. By accruing patients faster, we're going to get results faster, and that promising drug is going to help extend the lives of people like me faster, and that's exactly what we want. so we can help you. You are helping us with the research you're doing, and we're incredibly grateful for you, to you for that, but we can also help you. So ask us. It's a simple matter of asking. You know, all of you that are at big centers, a lot of you have patient advocacy groups, and you just have to reach out. I think I have my Twitter handle on every slide. I'm at Stage 4 Kelly. Tweet, you know, if you want to do a trial, tweet to me. I bet I know someone in your community. And again, you don't have to have an advocate that's 5 minutes down the road from you. You can utilize people that are all over the country to help you design a better trial. I love chemo brain, and I don't remember what I'm doing next. So here are also some systemic changes to improve clinical trial enrollment, not that just you can do. Again, inform every single solitary advanced and metastatic patient about the option of the trial in the first or second visit. I personally think that we need a centralized live person on the other end of the phone trial matching service that's funded by government, by industry, by academia. The Leukemia and Lymphoma Society does this. And they do that through philanthropic funding. They do it through some grants that they have. And if they can do it for these blood cancers, then why can't we do it for solid tumors? And again, industry, you are making a shitload of money off of us, so let's invest some of it back in getting patients enrolled in trials by having a live, real person answering questions and helping to match you with the service. I'm a doctor, and clinicaltrials.gov is challenging for me to negotiate, much less the average patient in rural Kentucky who has a high school education, or in Humboldt, California, where they're all like high on pot. Partner with your community oncologist. Let's bring the trials to the patients. Leverage technology. There's this like really cool thing called telemedicine, and if I can FaceTime with my daughter when she was studying abroad and she was in China, then I think that we can have the community telemedicine into the mothership institution for the informed consent process. Maybe use the clinical trial nurse at the mothership via telemedicine to go over the informed consent process with the patient. Because, you know, anybody in clinical practice knows that time is limited. Let's utilize our other partners, and let's utilize technology. Most labs and imaging should be able to be done locally in a CLIA-certified lab. Yes, there may be some very specialized tests that are only run at the mothership, but there's this cool thing called FedEx. If I can live in South Lake Tahoe, California, and I can get on my phone right now and order something from Amazon, and it will be there tomorrow, because they do delivery on Sunday now, which is totally cool with that big book I want to read, then we can utilize the same sort of thing to bring trials to the community and make it so more patients are able to enroll. Community oncologists and nurses are not stupid. You may be at that big, fancy academic institution, and you may have run 12 clinical trials, but my local community oncologist is also smart, and those certified oncology nurses are even smarter. Give them a protocol. Yeah, am I suggesting that someone do CAR T in Truckee, California? No. But there are a lot of other things that can be done where the patient does not have to go. If you are doing some of the cool imaging work with some alternative tags for PET scans or 2-micron CT scan slices that may not be available everywhere, okay, as a patient, I'm willing to drive occasionally to the mothership, but please don't ask me to do what someone at MD Anderson asked a patient to do, which is to drive 6 hours round trip for a routine blood test that could have been done in that patient's local community. And enrollment criteria should be broad enough that the trial population should reflect the patient that's going to be using the drugs. The FDA has published work recently on this issue and has said that we should no longer exclude patients for brain mets just because they have brain mets. We shouldn't be excluding people with HIV. And they're currently working on prior lines of therapy, washout periods, and a couple other criteria. Let's make the trials reflect the patient population that's going to be using it. And just to remind you, this is why we do research. These are friends of mine, dead and alive. Beth's not here today, but she made the slide. And in that lower right-hand corner, that's the reason why I want you to research. Because that's my daughter. And she graduates from college next year. And I want to be there because dead or alive, I'm paying for the party. So keep doing the work you're doing so that we can all be here for our families. Thank you.
Thank you very much. That was wonderful. And that rubber meets the road. Okay, I'm in good shape. Thank you for all that you do, the advocacy for patients and their families, the group here. It's critically important. The hematologists, oncologists, physicians and nurses need your help in educating and optimizing care of our patients who are contending with cancer in general. So I am very fortunate to be able to talk to you today about ongoing revolution in cancer therapeutics. That's not hyperbole. It's a reality. Buckle up your seatbelts because we're going to move pretty quickly or I'll get pulled off the stage here. Uh-oh. It says turn on something there. Okay, thank you, Dan. So this remarkable story begins from my perspective, my biased perspective. Right here begins right here with allogeneic stem cell transplant. Allogeneic stem cell transplant employed to treat patients with blood cancers, principally bone marrow failure syndromes. These are graphic data from a large registry called the Center for International Blood Marrow Transplant Research and demonstrate the remarkable growth in the last several decades of allogeneic stem cell transplant, such that there are now tens of thousands of these procedures performed on a yearly basis. Allogeneic stem cell transplant was initially developed to optimize and allow for the maximally intensive radiation and chemotherapy administration to patients and be able to recover the patient. Maximally intensive radiation and chemotherapy increases the chance that you'll kill every last cancer cell. However, it inflicts significant collateral damage on rapidly dividing cells in your body, and the most rapidly dividing cells in your body are your bone marrow stem cells that generate all your blood elements in your entire immune system. And so that collateral damage would not be associated with survival unless you could rescue the patient with a stem cell infusion. In this case, in the context of allogeneic transplant, those are the donor. Now, what was curing the patients was the maximally intensive radiation and chemotherapy, right? Wrong. Not so fast. So these are data that were published now almost 30 years ago that suggested that there was more to the story. Something else was curing many of these patients, and it wasn't necessarily the maximal radiation and chemotherapy thought necessary to destroy every last cancer cell. And that something in this lower curve is depicted in the context of a problem after allogeneic stem cell transplant called graft versus host disease. So the middle curves, if a patient did not develop graft versus host disease after transplant, their relapse risk for acute myelogenous leukemia would be about 30%. But if they develop this complication called graft versus host disease, their relapse risk was statistically significantly much less. My former boss at Memorial Sloan Kettering, Richard O'Reilly, said, geez, we know T cells cause graft versus host disease, and so let's develop a methodology whereby we take the stem cells at the lab and take out those T cells to prevent graft versus host disease, and it was very successful. We decreased the risk of graft versus host disease to less than 5% from 50%. But look what happened to the risk for relapse in these patients. The risk for relapse went up very significantly. And finally, you'd think an identical twin would be the optimal donor for a patient with leukemia undergoing transplant. They can't get graft versus host disease. However, the risk for relapse was much greater with an identical twin as opposed to a brother or sister who was HLI identical but not an identical twin. All of this suggested that there was something about the stem cell graft that was mediating potent anti-tumor effects. That something was in the T cell compartment, and we needed to learn about the biology of those T cells and leverage that knowledge so that we could optimize outcomes for our patients, and maybe we wouldn't have to give maximally intensive radiation and chemotherapy that exacts a significant toll on our patients. And so here's a patient several years ago, 5 years ago, that we treated with a leukemia, chronic lymphocytic leukemia, extremely high-risk disease, who undergoes a matched sibling donor stem cell transplant. And what you're looking at on the left-hand column is a biomarker, a PCR analysis of that patient's tumor cells, very, very sensitive. And on the right-hand y-axis of this curve, the percentage of cells that are donor. We want our patients to be 100% donor after transplant. Not to be 100% donor, it has a high association with risk for relapse. So look what happens to this patient. We do the transplant a couple months after transplant, that biomarker starts going up. This patient is going to have a frank relapse. So we give the patient an antibody therapy indicated for this disease. The patient has a transient response, but then several months after that, the biomarker shoots up like a rocket. The patient still has some of their own cells in their blood and their bone marrow. And so rather than giving chemotherapy or radiation therapy, we simply brought the donor in, collected lymphocytes from the donor's peripheral blood, and infused them into our patient. Those T cells attacked and killed the patient's cancer. The biomarker completely became negative, and the patient became 100% donor. And it's 5 years later, and this patient is still in clinical complete remission, demonstrating very clearly, and this is just one anecdotal example of many thousands of such examples, where we use T cells in this context, donor lymphocyte infusion, to cure our patients. At the same time that story was unfolding, Stephen Rosenberg at the National Cancer Institute was taking patients with solid tumors, taking their tumors to the laboratory and isolating, infiltrating T cells, stimulating them in the laboratory, and infusing them back into patients. This report from 1985 in the New England Journal of Medicine showed that a significant percentage of patients with melanoma had complete remissions, quite remarkable. And some of those complete remissions were very durable. One such patient who's a national spokesperson is 35 years out, having had metastatic melanoma in 1985 and part of this clinical investigation. Once it had become clear that T cells had the ability to control and kill cancers, a number of investigators started looking at, well, can we genetically modify those T cells to do an even better job? And that gave rise to the first genetic modifications reported now almost 3 decades ago as well. They didn't work, but proof of concept that you could start genetically modifying these cells, and perhaps someday it would work. And indeed, that has been realized. So we were left in those early years with this donor lymphocyte infusion. I failed to mention that the donor lymphocyte infusion is associated with significant graft versus host disease, which developed in the majority of patients, and some patients died from it. Stephen Rosenberg's work with those tumor-infiltrating lymphocytes, but the majority of patients don't respond, and even those who do respond have a high predilection for relapse. The lower panels are those patients who have genetically modified constructs in the modern era. And there are 2 such genetically modified constructs. It's actually even more complex than that. But in general, modifying the T cell receptor itself, which sees a cancer antigen on the surface of the cell, or generating completely new receptors that don't exist in nature outside of your genetic modification, called CAR T cells. And that's where we're going to focus most of our talk here. Now, the T cell receptor has to see parts and pieces of the cancer in the context of this heavy machinery called the HLA system. In contrast, the CAR T cells I'm going to describe can see anything that's out on the cell surface. Any glycolipids or proteins out on the cell surface can be targeted with CAR T cells. A little basic biology of T cells. I won't go too much into depth, except that the point of this cartoon slide is to show that dendritic cells, what's called a professional antigen presenting cell, that's a fancy way of saying these dendritic cells take up viruses, bacteria, tear them into pieces, and then put the pieces out on the cell surface and tell the T cells, if you see this, go after it, kill it in the future. And so that allows the dendritic cells to educate the T cells so they know that this is a bad actor, either a cancer cell, occurs in the same, very similar fashion, or an infection. But that T cell, stimulated by just that signal, will not become activated. It has to have a second signal. And that's why the first generation of these genetically modified constructs I showed you a moment ago didn't work, because it only provided one signal. The T cells have to have two signals. What do the T cells do when they receive those second signals? And this is really cool stuff. So on the lower part of this panel, this cartoon is a T cell, and in the upper part of the panel is a target cell, say a cancer cell. And so that cancer cell will have antigen presentation in itself and put parts and pieces of its own unique repertoire of proteins out on the cell surface, and the T cell receptor sees that in the context of the HLA system, and it forms a tight connection with that cancer cell, called an immunologic synapse, but a tight connection. And then it releases molecules that punch holes in the cancer cell. That in and of itself is a problem for the cancer cell. But that's not all it does. It punches holes in there, and proteins called nucleases race through those holes and go in and chop up the DNA. The chromosomes basically fall apart in that cancer cell, and the cancer cell dies. And that is the major pathway by which T cells work. So how do we make CAR T cells, and exactly what are these CAR T cells that everybody's talking about in the press? This is a patient on the left upper panel who's hooked up to a machine called a leukapheresis machine, going to collect just like donating blood, except we concentrate the white blood cells, donates blood. That is sent to the laboratory. In the laboratory, the T cells are isolated, stimulated to expand, and then you use a shell of a virus, essentially, and load it with a gene cassette for this new receptor that's going to recognize the cancer cell. Load it into that virus. The virus is replication incompetent, so it's not going to grow and cause a viral infection in the patient. That virus then infects the T cells, releases its payload, gets integrated into the DNA, and now the T cells start expressing this protein out on their surface called the chimeric antigen receptor modified construct, or CAR-T. That's expanded, that population of cells, sent back to the clinic in a couple of weeks, and then infused like a blood transfusion into the patient. Those T cells sweep around the body, recognize the marker on the surface of the cancer cells, form that synapse, and start killing. And so on the left-hand side of the panel is the native T cell receptor, normal biology inside of every one of us this very moment. And then on the lower part, the tumor cell presenting a molecule that might be unique to that cancer cell, recognized by the T cell so that it will kill it. On the right-hand side of the panel is the CAR-T with this construct that I just described to you, the chimeric antigen receptor. It can be designed to recognize anything on the cell surface you want it to recognize, and it doesn't need the HLA system. So you can target it against any molecule that might be unique to a cancer cell. Okay. There we go. And so this is a close-up snapshot of the chimeric antigen receptor on the right-hand side. On the left-hand side is the normal T cell receptor that's in all of us, and its machinery that's associated with it, and the signal 1 and signal 2 over there on the side of it. And on the right side of this panel are the CARs. The outside is the upper part, the outside of the cell. That's an antibody fragment that will recognize anything you want it to recognize. You got to stick it into the cell membrane. And then on the inside of the cell are what are called the co-stimulatory molecules, providing that signal 1 and signal 2. Signal 1 gets the T cell revved up. Signal 2 tells the T cell kill, that to which we are attached. So what should we target? What could we potentially target in cancer therapeutics and demonstrate proof of principle that this therapeutic strategy can work for patients? B cells are a great population of cells to target. And this is a cartoon depiction of how B cells grow up from a stem cell in the bone marrow all the way out terminally through many steps of maturation to a terminally differentiated cell called a plasma cell, which when that becomes cancerous, that plasma cell is multiple myeloma. There are all these steps in between that happen either in the bone marrow or out in the blood, lymph nodes, and then back in the bone marrow. And certain markers, certain molecules are present on that maturation pathway. And one such molecule called CD19 is present from the distal, just past the stem cell in B cell growth, all the way out and drop just before that plasma cell. And lots of cancers occur in that pathway. If it's in the bone marrow, acute lymphoblastic leukemia, B cell. If it's in the peripheral blood, mantle cell lymphoma, Hodgkin's disease, diffuse large cell lymphoma, follicular lymphoma, and then something called Waldenstrom's macroglobulinemia. So you could if you targeted CD19, you could conceivably attack any one of those cancers in the B cell lineage. And you can attack B cells and destroy them because you can live without B cells. B cells make terminally antibodies, proteins that help fight infection. But we can give patients intravenous immunoglobulin to keep them from becoming infected. You can't live with a chemotherapy refractory lymphoma, but you can live without B cells, at least for some time period. So B cells were the first targets of these therapies. These advances in cancer therapeutics with T cells made Science in 2013 Advance of the Year Cancer Immunotherapy T Cells on the Attack. In addition, just this past year in 2018, the American Society of Clinical Oncology called T cell immunotherapy the Advance of the Year. Why all the hoopla? There are two diseases I'm going to talk about briefly with spectacular results that have been durable in the majority of patients. The first is acute lymphoblastic leukemia. What happens to patients with relapsed acute lymphoblastic leukemia that's not responding to chemotherapy? These are the survival curves. It looks like advanced pancreatic cancer. These patients have survival numbered in weeks to months, rarely years, and only if I can get them back into remission and take them to transplant do they have a shot at long-term survival. What if I do a transplant on them and they relapse after transplant? These are the survival curve for those patients. These patients survive for very, very short periods of time. Again, weeks to months, rarely years would these patients be expected to survive. These patients demonstrate that CD19 molecule on their cell surface that I just described to you, and we're targeting with CAR T cells. In this very important study published in the New England Journal of Medicine last year, 75 children, adolescents, and young adults. Now, 61% of these patients had relapsed after stem cell transplant. And look at the complete remission rate, 81%. The overwhelming majority of patients went into complete remission. We now have follow-up that is greater than 3 years in these patients, and the majority of patients who went into complete remission are in continuous complete remission. And acute lymphoblastic leukemia patients who are going to relapse are going to do it in the first year or two, rarely beyond that. What about adults, true adults? These are pediatric and adolescent data. Here is a report from Memorial Sloan Kettering Cancer Center this last year in the New England Journal of Medicine. 53 adults, a little bit over a third of whom had relapsed after an allogeneic stem cell transplant, 83% complete remissions in those patients. That is absolutely unheard of. Those are stunning results. What about the most common form of non-Hodgkin lymphoma, diffuse large cell lymphoma? These are data from several large cohort studies, over 600 patients, showing what's the expected survival in this population if they are relapsed and not responding to chemotherapy. And the bottom line is a median survival is 6 months, and 7% are expected to have complete remissions to any subsequent therapy. So in that context, these 3 trials, published within the last year and a half, demonstrated with CAR T cells targeting that CD19 molecule, complete remissions of 50% rather than the 7% that I just showed you a few moments ago. These are not non-toxic therapies. These patients are taken care of in specialized centers around the nation. and have significant toxicities that we are contending with, increasingly successfully so, but we have a lot of work to do in that regard yet. Now here is an example of a 68-year-old who had failed 7 different types of chemotherapy regimen. Talk about being disheartening, and with a lot of associated medical problems. So a patient of advanced age, we discussed some of that earlier, in fitness for chemotherapy. This is a PET scan in this patient. All of the black stuff is bulky tumors. This patient had 15-centimeter abdominal tumors, receives CAR T-cell therapy, and this is this patient's PET scan 30 days later. So complete normalization. This patient continues in complete remission after CAR T-cell therapy. That's an anecdote, but these complete responses occurred in 50% of patients. Multiple myeloma, survival curves for patients who have relapsed disease after their primary therapy. Not an attractive picture. And so in that context, this small study, 18 patients with multiple myeloma, received CAR T cells, 100% of patients responded. But initially, only 27% went into complete remission. But without any other therapy, as you followed them over the next several months, increasingly more and more people kept going into complete remission. Why? This is not a drug that goes away and is metabolized in your liver or your kidneys. This is a living therapy. These are your cells. They're still around months later, and they're still doing their job. And they're still fighting cancer cells for months, perhaps even years, after they've been administered to patients. That's pretty remarkable. These are data for acute myelogenous leukemia, the most common form of acute leukemia in adults. We heard earlier about acute myelogenous leukemia and fitness for those patients. Its survival is extremely poor in patients who relapse. What can we target in acute myelogenous leukemia that's a molecule on the surface of the cells that's not present on normal healthy cells? There are a number of different molecules that we could consider targeting. However, those cells are also either back on the stem cell, you can't wipe out all the stem cells unless you transplant the patient, or they're on cells that are absolutely essential to survival, in contrast to the B cells I described earlier. If you give the CAR T cells and you wipe them out, you can say, well, we can do a stem cell transplant now on this patient and rescue them. But you can't, because the CAR T cells aren't living therapy, they're still there. So you've got to be able to turn those CAR T cells off. You need a light switch, an on-off switch. Such a switch has been developed without going into too much biological detail here, whereby we can give an inert dimerizer and cause these cells to undergo death when we're done with them. When they've killed all the cancer cells, shut them off. So we can treat diseases like acute myelogenous leukemia. We have a toolbox now, a repertoire. What's remarkable is those complete response rates of 80% acute lymphoblastic leukemia, 50% relapse refractory large cell lymphoma. Those are stunning results. This is just the beginning. So I do envision a future where we're going to see the days of conventional non-specific chemotherapy be numbered, and conventional non-specific radiation therapy, and be much more specific in our therapies. And I think that this is part of that broad story in cancer therapeutics. And finally, this slide demonstrates a clinical trial that we're opening currently, whereby rather than waiting 2 weeks, 3 weeks, some patients will die during that period. These are aggressive diseases. Rather than waiting that time period to make their own cells, have third-party cells available to them. How can we do such a thing? They'll be rejected or cause graft-versus-host disease. We can use a technology called CRISPR-Cas9 and edit out the T-cell receptor that would cause graft-versus-host disease, and edit out the HLA system that would cause rejection. And we are beginning enrollment on such a multi-center study in our institution. So it's a brave new world, extraordinary developments coming down the line. These phase 2 studies, because of the results, led to the first Food and Drug Administration approval of any genetically modified cell therapy. And they were based on phase 2 trials, not large prospective randomized phase 3 trials, because of the compelling outcomes of these patients. Thank you very much for your attention.
Good morning. First of all, I'd like to thank the NHL. This is a great opportunity to come talk to you guys. I've had some great discussions. I've really enjoyed this conference. If you haven't figured out already, I was originally scheduled to talk in the organ-specific metastasis, the dormancy, the bone metastasis, brain metastasis, and switch the schedule around a little bit. I'm going to shift gears a little bit from the clinical trials to shift to some of the earlier inception of these studies, the bench science on how these projects get started. And I thank Kelly for making the point that advocates, I don't think we probably don't need to tell this group, but I want to stress advocates need to come into a project at inception, not 4 days before a grant is due. And Jenny, we just had that discussion that, hey, I have a grant due in 4 days, can you help out on this? And well, I want to know something about the project. And so I want to talk a little bit about some of the inception, and then how it relates to how we design clinical trials. And I hope at the end it'll become relevant to everyone. So here are the objectives, and we've heard a lot about bone metastasis, and so I don't really need to go in too much depth about that. But I want you to focus on that third point, understanding therapeutic strategies, and how we can think about treating patients, and then how that relates to designing clinical trials. There is a delay. Sorry. And so we've also heard a lot about Stephen Paget's work from the late 1800s, where he realized that the distribution of metastasis is not just based on blood flow, and there's a prevalence to bone metastasis that's not easily explained. And we often hear this statistic that, well, up to 70% of patients with metastatic disease have bone metastasis. And there's been a lot of studies looking at the distribution of metastasis to show that maybe that's true. And there's one that I point out here. 73% in this case, 73% of patients have bone metastasis. I want you to be careful about these statistics. What do they actually mean? Because a number of other studies show that, yes, bone is prevalent, but maybe it's 50%, maybe it's 49%, maybe it's 60%. And some studies even show that bone may not be the most prevalent. Point being is that bone is very prevalent, and we need to really think about it and develop targeted therapies specific for bone, and just not get too wrapped up into the statistics and how they were generated. One of these studies by Shi Wei also showed that it depends on subtype of breast cancer, and so luminal breast cancers, those that are hormone negative, predominantly go to bone, but the HER2 positive and the triple negatives, that's not the most prevalent site. So we have to consider subtype of breast cancer when we think about where they go to. Just as a side note with statistics for this group, we always hear that 90% of patients that die of cancer, they die from metastasis. Where did that statistic come from? I think we'd all agree that it's probably true. I looked that up. I tried to find it. It's really difficult to find a study that actually shows that statistic. Why isn't it 95%? Why do we say 90%? Is it 83%? No. Look it up and see if you can find an actual statistic. It's kind of interesting. But without getting too wrapped up into the numbers, I think we'd all agree that for breast cancer patients in particular, bone is a prevalent site. And I want to talk a little bit about that, the bone environment. Now, we've already seen a figure similar to this. Both Connor and Erwin had shown and introduced this concept of the bone environment. There's a lot of cells that make up the bone niche, and as we heard yesterday, there's niches inside of the bone that are not all the same. But when we think about the bone environment, we often talk about the bone-forming cells, the osteoblasts, and the bone-degrading cells, the osteoclasts. And as I said, they're not the only cells, but those are the ones that we tend to focus on the most. And there's a balance between those. As we develop, as we get older, the bone is constantly remodeling. So these two cell types are very important, and they regulate each other. There's both bone degradation as well as bone formation. It's very important for healthy bone. So what happens when cancer cells arrive in the bone? I know this is a little bit busy. I really like this figure. It was taken from Andrew Master's group. She wrote a review in 2010, and I think it's still very applicable today. And it shows very nicely that when cancer cells get to the bone, they're in communication with these host cells. They communicate with both the osteoblasts, the bone-forming cells, as well as the osteoclasts, the bone-degrading cells. And sometimes we hear about the vicious cycle. What is this vicious cycle? It's really a complex series of events. The breast cancer cells tend to communicate with these bone-forming cells and basically tell them to communicate with the cells that are responsible for making the bone-degrading cells. They secrete things like rank ligand that I'll talk about in a moment. This stimulates the formation of the bone-degrading cells, which then degrade part of the bone and release factors that are found inside the bone, cytokines, growth factors that then stimulate the cancer cells to stimulate them to continue to proliferate. And of course, that's put in very simple terms. There's a lot going on in addition to this, but for the most part, this holds true. How do we target these? How do we target this environment? Bisphosphonates have been used. You probably have heard about this. Bisphosphonates stick to the bone. They inhibit the bone-degrading cells, the osteoclasts. They tend to promote the death of those cells, apoptosis. And in addition to the bisphosphonates, a bit more recently, we have denosumab. Denosumab is an antibody that interacts with rank ligand that I just told you about, that inhibits the formation of the bone-degrading cells. And where that's advantageous is if you inhibit the bone-degrading cells, then you at least stop this so-called vicious cycle. And that is FDA approved, but then, and there's lots of other targets for the osteoclasts, for the degradation of the bone. But then the other approach is to stimulate the osteoblasts, those that are responsible for forming the bone. And for the case of breast cancer metastasis to the bone, we often think about the targets that are already in use for osteoporosis. And one such is this teriparatide that is a mimic of the parathyroid hormone that stimulates osteoblasts. Now, this is a bit controversial because long-term use has been associated with osteosarcoma. So it may not be something that we'd use in the case of bone metastasis. But these are some of the strategies in the case of bone metastasis is to target these host cells and disrupt this vicious cycle, in addition to targeting the cancer cells themselves, as we heard yesterday from Erwin. So what I want to do now is just give you an example of why it would be important to understand some of these strategies as we sometimes put our blinders on and we look at exactly how these molecules are impacting our cell of interest, in the case of denosumab or something related, that we're just focused on these osteoclasts, these bone-degrading cells, and we sometimes forget about what is that doing to some of the other cells, because it may be quite relevant. So one of our projects has to do with this SYN3 chromatin complex. We heard earlier this morning this term epigenetics. I'm not going to go into all the background of why we're focused on this, but what we found was that this SYN3 complex is very important. In fact, it's necessary for osteoclast formation. I promise I don't have too many data slides. I think I had 2 or 3. But without knowing what this is, you can see a difference between this panel and the panel below it. These are osteoclasts that we've grown in vitro on dishes, and you can clearly see a difference when we lower the levels of this SYN3 gene. We do not form fully functional osteoclasts, and we can see it's statistically significant. So, in addition to doing these in vitro assays, we can look at mouse models where we turn off this gene in the precursor cells that make osteoclasts, and when we do this, we see double bone volume in the mice. You can see that right here. The bone volume is increased because we have a reduced number of the osteoclasts, those that degrade the bone. So we're very interested in this molecule and how it impacts the bone-degrading cells. But in addition to that, this gene is being targeted for its ability to inhibit breast cancer cell invasion and metastasis. So this is kind of complicated, but I'll kind of walk you through this. Is that for 20 minutes or is that for 12 minutes? Because I promise I'll be wrapping this up soon. So, for those of you who are not familiar with looking at protein structures, this is just a linearized view of SYN3. And what this is it's a scaffold protein. It doesn't have any intrinsic activity on its own. It doesn't bind to DNA and turn genes on and off by itself. I mentioned epigenetics. It doesn't modify the structure of how DNA and proteins are interacting and how DNA is packaged. But it recruits these other proteins, and those proteins are what does its work for it. And what I want to focus on here is that Sam Waxman's group at Mount Sinai has developed this SYN-interacting decoy peptide that prevents the binding of an important factor that regulates how genes are turned on and turned off. These are known as transcription factors. And found that by doing this, you can inhibit breast cancer cell invasion and tumor progression in vivo. And then had this whole drug screen to show that small molecules that mimic this peptide can be useful in a preclinical setting. And so what we did is we said, okay, well, maybe in these breast cancer cells, this scaffold would be necessary in order for the cells to metastasize. And this is where it got a little interesting, because SYN3 is not just one protein. It turns out that it has a brother. There's SYN3A, there's SYN3B, they're very similar. And when we reduce the levels of SYN3A, we see a very invasive breast cancer cell. But when we reduce its brother, SYN3B, we get these colonies that look more like a normal breast cell. They're not invasive. And this holds true with invasion assays, and when we look at metastasis, we see an actual increase in metastasis when we reduce SYN3A, but we see a decrease when we reduce SYN3B or when we target both. And so as we look at how we could potentially target this, this is sort of a summary of some of the next studies. We know that if we inhibit the binding of these transcription factors to the scaffold, that we can reduce cancer cell invasion and metastasis, but that's because we're not just targeting SYN3A, we're targeting both of these scaffolds. When we specifically reduce one, in the case of SYN3A, we actually increase invasion and metastasis, and when we reduce SYN3B, we inhibit. So we have to understand what our compounds are doing and how they're targeting something like this. And we can talk whether or not this would be a good target or a bad target to go after. There are many reasons why we wouldn't want to go after this in the drug setting, but it's an interesting example of how understanding the biology is very important. And I'll give you one more example. I put this at the bottom. Know your target and how it affects different cell types, whether the host cells, the osteoblasts versus the cancer cells themselves. We have to understand this. And this seems very logical, very basic. We should all know this. But I want to step back. In the case of matrix metalloproteases back in the 1990s, these were being pursued as targets, and they rushed into the clinic. All the inhibitors of this family of enzymes, the MMPs, rushed into the clinic, and guess what happened? The trials did not go like they predicted, and in fact, they were not successful. Well, what did that do? It really set a whole field back, and some of you work in this field know that when those trials were not successful, everybody that was submitting grants on those particular enzymes were not getting grants funded because everybody said, well, clinical trials didn't work, so this is not a good target, so we don't even want to study it. And that's a shame because there's a lot of important biology and relevance to the MMP field. And I see some of the same things happen. I have up here the HDAC inhibitors. You know that some of the HDAC inhibitors are FDA approved for lymphoma. They haven't fared well in solid tumors, and because there's a lot of complicated biology that goes with this. And so understanding a little bit of what your target is doing is very important when we set up clinical trials and setting them up correctly so that we don't have a whole field that gets set back. So I am wrapping it up now. This is my last slide. I hope that I can illustrate that bone metastasis is prevalent, and that it's not just the cancer cells that get to the secondary site, but it's the host cells as well. And if we think about yesterday's session with the brain and the bone metastasis, the lung metastasis, not all of these metastases are identical. And understanding how the microenvironment, those host cells, play a role in communicating with the cancer cells is going to be very important for how we design trials and how we treat this disease. Not all metastases are the same. And so I hope you get that point from all of this. I just want to acknowledge, did get funding from METASTATIC several years ago, and we recently got a grant funded, and I want to thank, in particular, Thelma Brown. She has been an advocate for a lot of our studies, and she is one that believes that taking part in a study at inception is very important so that as the study progresses, they can be an active participant in all aspects. So with that, thank you for sticking around, and we now get the pleasure of hearing from Danny Welch.
Thank you all. I also, before beginning my actual presentation, I want to give a big shout out to the AV folks, Anna, Spencer, Jordan, and those guys. They've really helped losing a computer board and stuff. They've helped us get all this stuff set up. And thank all of you for being here. Thank you, Sonia, Kristen, Kate, Jill, everybody who's been here. It's been a fun time, and I hope you've enjoyed it as much as we've had. So my task this morning is to talk about designing clinical trials. And the story actually starts when I finished graduate school. I took a job in the pharmaceutical industry and started, I was given the opportunity, should we work on as a company? And I suggested that metastasis should be the target. And it was the first and still the only drug company that actually had a full metastatic anti-metastasis program that focused specifically on targeting metastasis. There is currently no company that does that. Largely because every pharmaceutical CEO says everything we do is for the metastatic patient, which, shall I say, is bullpucky. So what I'd like to do is deal with that issue, but discuss the challenges and opportunities that are associated with designing targets or trials, specifically targeting metastasis, and help us begin the thought process of how to implement those trials and to identify what the appropriate outcomes are. That's the purpose of this meeting. What are the outcomes that we are looking for? So we have to have a few premises. Doug is right. Finding an actual study that shows 90% of morbidity and mortality is attributable to metastasis is something that everybody can see, but nobody's measured in an explicit way. If we are going to have to move the needle in terms of cancer survivorship, we have to either prevent and treat metastases. I think we can all agree on those premises, but the two slightly more controversial premises are that metastasis is a viable therapeutic target. No one in this room thinks otherwise, but there are people who do think otherwise, mostly because they say the horse has already escaped the barn, why do we need to target metastasis? And the other premise that I hope I can convince you of in a couple of minutes is that targeting the primary tumor is not the same as targeting the metastasis. And I think Chad was starting to introduce that yesterday, but there are specific reasons for saying that. First, what is our objective? It's either to prevent or treat metastases, but it's also both quantity of life coupled with quality of life. Frankly, I don't want to live if I can't have some quality. I think most of us are that way. We need and want to have a purpose. So we want to check off those boxes. If we are to do that, we have to identify a target. And what is the ideal target? The target has to exist on every metastatic cell so that we can target all metastases. It should be something that's unique to the metastatic cell so that we have fewer side effects. Because if we target something that's elsewhere, the chance of a side effect increases. And the target must be accessible. It doesn't do us any good to have something to treat brain metastasis, as Josh was saying, that doesn't get into the brain to access the cell. Amazingly enough, I sit at the perch of the journal Cancer Research. I'd say 90% of the papers submitted talk about drug resistance, talk about molecular mechanisms in the tumor cell, completely ignoring that there are pharmacokinetics and pharmacodynamics, the drug getting to or from the target and getting into the cell. So to answer the question about what, if anything, is unique about metastases, Doug and I undertook this thought to identify what are the hallmarks of metastasis. A hallmark is something defined as existing in all the cells. And metastasis is something on top of making a tumor. So we start with, you make a tumor cell. But what makes it metastatic is not every cell in a tumor becomes a metastasis. And after reading more than 10,000 papers, most of which are boring as all hell, I would agree, we tried to critically analyze and define what we think are the hallmarks. The first is you've got to get up and run. So targeting the ability to crawl or the ability to degrade a tissue are legitimate targets. Plasticity. This one's tough to target. Plasticity means adaptability. A cell that metastasizes is in one place, it goes through, it's basically on the road through the bloodstream, lymphatics, or across the body cavity. That's a different environment. If you are traveling to Europe, using the analogies that Josh used yesterday, you have to be able to survive the dehydration that takes place on an airplane, or the seasickness that might take place on a boat, or if you want to walk the Bering Strait, okay, you can do that. But there are all of those different things that you'll encounter. The tumor cell has to adapt through all of those processes. So how can we make it less adaptable and maybe increase the killing that takes place in between? Doug just talked about how the cancer cells modulate the environment. They not only manipulate the bone, they also manipulate the lung, the liver, the ovaries, all these other tissues. And they're basically doing that by co-opting the mechanisms that already exist in the immune system and the other tissues. They're like sleeper cells when we think of terrorists. That's not politically correct, but I'm not politically correct. The fourth hallmark is colonization. And this is, I think, began the discussion yesterday during Chad's presentation. Millions of cells start the process, but very few actually make metastases. So all of these hallmarks can be targets. The problem is that mechanisms for each of these are still not well understood. So we've identified these targets, but the cancer cells, the metastatic cells, use exactly the same mechanisms that normal cells do. They're not reinventing things, they're just using them inappropriately. A lot like teenagers. They do everything inappropriately, but they're normal, sort of. Okay. Another big buzzword lately is tumor heterogeneity. Every tumor cell is a unique entity. It's like saying human. Look at the person next to you. They do not look like you. They do not behave like you. No one behaves as appropriately as you. Okay? But you're all human. So we can't lump all cancer cells as equal. They're all different. They're genetically unstable. The problem is during the growth of the cells, the manipulation, the DNA gets all mucked up and these cells accumulate those changes. A biggie for the pharmaceutical industry is the dissemination, which is distinct from metastases. Because flying around is not metastasis. It's a cell that's moved. But it has to grow somewhere else to be a metastasis. At least that's what Doug and I would like to convince the field to be clear with the terms. That occurs for diagnosis of the metastasis because you have to have a big enough mass to be able to see it. Each tissue is different. A metastasis in the brain is not the same as one in the lung or one in the liver. We have to treat them as individual types of metastases. And unfortunately, as well, there is biochemical redundancy. There's more than one road to Rome. You have to recognize if we put a roadblock here, the tumor cell, by that genetic instability, tries to find a way around it, over it, through it, underneath it, whatever. So if we want to design a trial, we've looked at the targets. That's obviously already still a challenge. But we have a few possibilities, blocking colonization as an example. Let's say we identify a target. What are we going to define as a successful outcome? If we have that outcome, it has to be measurable. And who's doing the measuring? Is it the patient, the caregiver, the government? Okay. All three of those may have different positive or negative outcomes. And we have to get on the same page. What are our barriers to success? And even if we can design a therapy, if it costs $1 million per patient, it's not feasible. So we have to have all of these for designing a clinical trial. Here are the outcomes that I sort of have come up with, and the things that would give us a reason to have a high five. I was told there's a study that says if you show pictures of cute pets, your presentation gets reviewed much better. That's why I'm showing it. Disease outcome. First, best thing is a cure. There's not a cancer cell remaining. That's the perfect desired outcome from a disease outcome. The second is that we render metastases a stable chronic disease, something, even if the cancer cells are there, they're controlled. It's not as good as getting rid of them, because if you leave a cancer cell, there's a potential it could grow again, but that would be a legitimate outcome. The disease outcomes involve shrinkage, the stable or growth of the metastases. We'll come back to that. And a measurement, the liquid biopsy. The markers go away. There's no evidence of circulating tumor cells or the DNA. The time to some recurrence. If every patient all relapsed in 6 months, and we gave a therapy that every patient took 5 years, is that a success? Time to death. If we can prolong life, that's a good thing. The challenges with recurrence time, which is currently one of the things that most people talk about, and I'm just going to use a couple of examples here, is we don't know how long it's going to take. If you have slow-growing cells that are already there, some can take years. There have been documented cases of recurrence in cancers 30 years after defining no evidence of disease. I mean, tumor cells have already disseminated, but they've done nothing. The patient lived a, quote, normal life, except for the skin anxiety types of things. But that is a good thing. But no clinical trial can ever last 30 years to call it a success. So we have to have something that's measurable and feasible in a certain amount of time. There's something known as the RESIST criteria, response evaluation criteria for solid tumors. RESIST, in shorthand, I am not a clinician, means you have at least 25% shrinkage, okay, and or disappearance. But those other outcomes that I mentioned, no new metastases, that would be a failed trial because you're not measuring something. If it doesn't happen, you can't measure it. The dormancy of metastases, if you prolong things and they're staying stable, that's not a success. It would be considered a failed trial or a stable biopsy or liquid biopsy. Those would be considered negative. So we have to identify new criteria for these trials to measure whatever the outcome we want or desire. Quality of life outcomes, these are issues that we talked about in the last session: mobility, exercise, vitality, quality issues, having a social life, limitations with work, the ability just to do those things that are involved in daily life. And those, I think, need to be measured alongside. I would consider it a success, not ideal, but still a success, if we do things that improve quality of life, even if the quantity isn't there. I think that's the corner of my eye. Oh, I thought you were looking at you at the hot dog. All right, so here are some of the challenges, and I just want to put them before you in terms of discovery and preclinical activities. There is nothing unique in a metastatic cell. There is, I've not heard of it. old enough and been doing this long enough that I would think I would have heard of it. Shockingly, not a single pharmaceutical company tests a preclinical drug in metastasis models, yet that is what we are trying to treat patients with in the clinic. Surgeons remove the primary tumor, but we're not testing drugs against metastases. We already know that metastases are not the same as primary tumors, and certainly not all tumors grow in the skin, which is how most drug companies test things. It's not relevant to where the tumors start. So they're doing it all wrong. There are reasons for doing it wrong. It's cheaper, faster, reproducible, and all of that. But is it relevant? Relevance to me is a high criteria. Another big issue that was part of your comments at the last meeting, there are too few models to assess efficacy. You can count the number of metastatic breast cancer models on one hand. Metastatic prostate cancer, you can count on one finger. Ovarian metastases, one or two fingers. Lung cancer, I don't know how many, but not many. And there are complications associated with that. We don't have the models. No agency will pay for development of new models. It's horribly expensive and usually fraught with failure in the interim. But if we don't start getting better models more reflective of what happens, we're not going to move the needle adequately. And unfortunately, this is my belief. Too many scientists jump onto the hottest bandwagon and say, this is better than the old ones. Every model is wrong. Every model can tell you something. You just have to know what the limits are in order to not overinterpret. I will now get off my soapbox for a moment and continue with that. All the models are highly complex. I think Doug can tell you from his sentence in my lab, I worded that appropriately, did I not? That to do a metastasis study well and do it reproducibly is very difficult. And all the lab folks here realize that, and it's really tough. And another is the regulatory burden for the Animal Care and Use Committee. Currently, my animal protocols are double the length of the IRB protocols for human studies. For mice. I have no desire in my life at all to torture mice or small rats or anything else, but those are the issues. They also question, we have to let, in order for the models to work, the tumors have to get fairly large so that we get them reproducibly. The IFIC says, oh no, you can't have a large tumor. So we have to write all these exemptions. The protocols are very burdensome. I had to give them 3,000 pages of documentation not to have to administer unnecessary drugs for a study. 3,000 pages. Trial-related issues. I talked about the RECIST endpoints, the time to the result. We can't have trials that last for 20 years. So we have to have something interim. Bob Earhart and I, when I used to work at Upjohn, actually got the first paper signed that biomarkers were a legitimate endpoint. Especially for metastatic disease. That was signed in 1984, but the first biomarker tested was not until the late 1990s. So we knew that would have to be, but it still took time. The complications for an anti-metastatic therapy will be complicated by the primary tumor therapy. Drugs interact. And there are survivorship-related issues associated with every chemotherapy agent, neuropathies, chemobrain. They're just a couple. The regulatory burdens of the IRBs, those are all there. And currently, metastases are not recorded in all of the SEER databases. It is something different. As patients, you need to get that changed. That's important that legislators do that. Oh, really? All right, patient-related issues. I am not a metastatic patient, but I've talked to many of you, and hopefully I've addressed a few of these. Kelly talked, patients have to be involved in the trials and in the consent. and that's critical. Patients go to the trial instead of the trial coming to the patient. That's probably a big issue related to low participation, especially in underrepresented or poor communities. Enrollment in trials seem only to occur after other options are exhausted. That is not palatable to me. There are conscious and unconscious biases. Unfortunately, too many people diagnosed with metastatic disease, many of their friends, well-meaning, don't want to say the wrong thing. And because they equate metastasis with dying. So they don't want to say the wrong thing, so there's a bias. I don't think many people want to behave that way, but I'm sure I even behave that way. And we need to understand it. Financial toxicity, the inability to work, all the difficulties that are associated. You all know them better than I do, but those are some of the challenges that are facing us. So with that, I thank you for your attention, and I love the talk of this. Now you can clap. And